hear me? Okay, so we start with the lecture 6. And now we are already moving to specific bacteria that are causing different infections. And first of all, we will start with the cocci type bacteria. You remember that these are round bacteria, sometimes elongated. Some of them are gram positive, some of them are gram negative. So we will go through those different species that are interesting from medical microbiology point of view. And this is, of course, Staphylococcus, Streptococcus, Neisseria, and so forth. You can see the content. Okay, let's start with the classification of this bacteria. All of them belong to the family Micrococcaceae, and for us, genus Staphylococcus and Micrococcus and the species Staphylococcus aureus, Apophyticus, Epidermidis are more important than the others because they are associated with different infections. But actually there are many other species included into this family, and among them we can name, for example, Micrococcus luteus. There are many others. So Staphylococcus were discovered by the Scottish surgeon whose name was Sir Alexander Oxton. In 1880, he was trying to study these bacteria under microscope, and he found out that there are cluster-forming microorganisms that were associated with different diseases. Pyogenic diseases. This means that pus was developed due to these infections. And he was the one who introduced the name for these organisms, Staphylococcus. This name comes from Greek, staphyl, which means bunch of grapes, and coccus, which means grain of berry. Grain or berry. So these Staphylococcus are facultatively anaerobic bacteria. There are catalase positive and gram positive. So these are those characteristics that we should remember. You know very well already what this means. Facultatively anaerobic means that these bacteria can survive in anaerobic conditions. Catalase positive means that they can digest, they can produce catalase, and gram positive means that their cell wall is has certain structure. The murine is included into the cell wall. The genus Staphylococcus contains about 40 species and many subspecies, but as I already mentioned, Staphylococcus aureus is the leading cause of different infections. However, during the last probably 2 decades, Staphylococcus epidermidis starts to dominate in the clinics and is causing so-called hospital infections. So this shows you the structure of Staphylococcus. Again, these are gram-positive cocci. They are non-motile. They are facultative anaerobes. Cells, you can see that they occur as clusters. And this is because the cell division occurs along the different planes, and the daughter cells remain attached to one another. What it is interesting about these bacteria, they are salt tolerant. So this means that they can survive in salt, which is present on the human skin. And this is one of the properties of the pathogenic bacteria. Also, these bacteria are tolerant to desiccation, and this allows them to survive in environment on the surfaces like formities, for example. And this is why these bacteria became so dangerous for the hospitals and causes nosocomial infections. Staphylococcus can produce this polysaccharide capsule rarely. The peptidoglycan layer or murine is the major structural component of the cell wall, and it is important in the pathogenesis of the staphylococcal infections. Other important component of the cell wall is teichoic acid. It's not working, I don't know why. Okay, you can see, I think that you can see. Okay, teichoic acid is here. Okay, I think that I should move probably. Okay, teichoic acid is here, and peptidoglycan layer, you can see that it is very thick. This is why we have blue color after gram staining. Half of the cell wall by weight is formed by peptidoglycan. This is a feature which is common for the gram-positive bacteria. And the subunits of peptidoglycan, they are N-acetylmuramic acid and N-acetylglucosamine, NAM and NAG. And unlike gram-negative bacteria, the peptidoglycan layer in gram-positive bacteria consists of many cross-linked layers. You can see them here. See? So this is NAM and NAG, and they are connected, these layers are connected with these peptides, and there are many cross-linked layers. Protein A is the major protein component of the cell wall. It is located on the cell surface, but it is also released into the culture medium during the cell growth. And the protein A has a unique property because it has an ability to bind to the Fc part of all immunoglobulin molecules except immunoglobulin G3. And it is not an antigen-antibody specific reaction. The surface of most Staphylococcus strains is uniformly coated with protein A, but there is an exception. Coagulase negative Staphylococci do not have the protein A surface on their surface. The presence of protein A has been exploited in the serological tests, which uses this protein A coating, and Staphylococcus aureus is used for Staphylococcus aureus, but this is a non-specific carrier of antibodies directed against other antigens. Additionally, detection of protein A can be used as specific identification test for Staphylococcus aureus. So it distinguishes Staphylococcus aureus from Staphylococcus epidermidis, Staphylococcus hominis, and other staphylococcal species. So you can see here the binding of protein A. Okay, so you see protein A is looks like yellow, some yellow appendages here, and it can bind to Fc portion of immunoglobulin. And if it binds here, in this case it means that it cannot bind to the Fc receptors of the phagocyte, and phagocyte cannot engulf Staphylococcus and cannot digest it. So immunity is not working in this case. If we are talking about Staphylococcus epidermidis, so you can see that there is no protein A around the cell, and of course it is not binding to FC portion, and this is why the Staphylococcus epidermidis phagocytosis is not blocked. It is easily phagocyzed by the phagocyte. Here is antigenic structure of Staphylococcus. As we already mentioned, capsule is very rarely formed, but it can be formed by some species. Or instead of capsule, a polysaccharide slime layer may be formed as well. So you can see that here we have the peptidoglycan layer. Here we see the polysaccharide A or teichoic acid, and this is protein A. Deeper in the cell we see cytoplasm. We see also in the cell wall we see the clumping factor as well. So what is the significance of teichoic acid? Teichoic acid is species specific, phosphate containing polymer that are bound covalently to the peptidoglycan layer or through lipophilic linkage to the cytoplasmic membrane. So the lipopolyteichoic acid is abbreviated as LPA. Teichoic acid mediates the attachment of staphylococci to mucosal surfaces through its specific binding to fibrinectin. Coagulase is another protein. The outer surface of most strains of Staphylococcus contains clumping factor, which we already saw on that picture. It is also called bound coagulase. This protein binds fibrinogen, converts it to an insoluble fibrin, causing the Staphylococci to clump or aggregate. Detection of this protein is the primary test for identification of Staphylococcus aureus, as well as identification of A protein. So I think that you have seen this already. This is one of the tests for identification of Staphylococcus aureus. It is a coagulase binding test. Coagulase, as I already mentioned, causes blood aggregation, coagulation. Blood clot protects the bacteria which is inside the clot from phagocytosis, and also provides different defenses for these bacteria. Staphylococci are often coagulase positive. So we can see here the test. So this is serum in this tube, and if we inoculate Staphylococci into this serum, we get aggregation of these proteins and pelleting them. This means that this particular strain is coagulase positive. If the serum remains transparent or remains evenly turbid, in this case this means that Staphylococcus does not have coagulase activity. So it is coagulase negative. The same test can be performed on the slides. Here you can see the positive outcome of this test. You can see the aggregations. And here, since we do not have any aggregations, this means that the outcome is negative. So only Staphylococcus aureus is positive, while, for example, Staphylococcus epidermidis, hominis, saprophyticus, and others are Coagulase negative. As I already mentioned, Staphylococcus epidermidis becomes more and more spread in the hospitals. So one of the initial tests that should be done, it is a coagulase binding test to distinguish between Staphylococcus aureus and Staphylococcus epidermidis. So other surface proteins that appear to be important for adherence to the host tissues include collagen binding protein, elastin binding protein, fibronectin binding protein. As to capsule, capsule is formed rarely. It can form capsule, some of species can form capsule or polysaccharide slime layers. Polysaccharide slime layer is a loose-fitting polysaccharide layer. It can occasionally be found in staphylococci which are cultured in vitro, but it is believed that in vivo probably this kind of developments are more frequent. 11 capsular serotypes have been identified in Staphylococcus aureus, with serotypes 5 and 7 that are associated with the majority of infections. So you can see here, this is A protein here, clumping factor, and the capsular polysaccharide. This is highly capsulated in this case. It is poorly capsulated. If it is poorly capsulated, then we have the slime layer. Like in other cases, capsule also helps Staphylococcus to survive and to avoid the immune system reaction. The capsule protects the bacteria by inhibiting the hemotaxis and phagocytosis of Staphylococci by polymorphic nuclear leukocytes, as well as by inhibiting the proliferation of mononuclear cells. It is also facilitating the adherence of bacteria to catheters and other synthetic materials, which is a very big problem today in the hospitals, because these devices are becoming the sources of infection. Instead of helping patients, we are making harm if this bacteria is forming is adhering to the catheters or other synthetic materials. By the way, you know, these artificial joints also may be infected with Staphylococcus, and in this case we can get serious problems as well. And this is because they can form biofilms on the surfaces of these different devices, medical devices, also heart valves, for example. Now about cytoplasmic membrane. The cytoplasmic membrane is made up of the complex of proteins, lipids, and small amount of carbohydrates. It serves as an osmotic barrier for the cell and provides an anchorage for the cellular, biosynthetic, and respiratory enzymes. And among the enzymes, we already know that there is coagulase, catalase, hyaluronidase, fibrinolysin, lipases, nucleases, penicillinases. Penicillinase is also known as lactamase. Last time we were talking about lactamases that are destroying antibiotics. Now, about the role of different enzymes. Coagulase, you already have heard today that it is triggering blood clotting. Hyaluronidase breaks down hyaluronic acid, enabling the bacteria to spread between the cells, and this means that it is one of those enzymes that is helping bacteria to cause systemic infection. Staphylococcus, in other words, it is fibrinolysin. It dissolves fibrin threads in blood clots, allowing Staphylococcus aureus to free itself from clots. Now, I think that you paid attention that there is a coagulase activity and an opposite activity, Staphylococcus activity. Coagulase activity helps the bacteria to survive inside the clot and to propagate inside this clot. And when there is a big amount already accumulated, in this case, they start to produce Staphylococcus, another enzyme, and free themselves and are released into the bloodstream and start to infect different remote parts of the body. Lipases. These are digest lipids, allowing Staphylococcus to grow on the skin surfaces and its cutaneous oil glands. Beta-lactamase or penicillinase breaks down penicillin, allows the bacteria to survive treatment with beta-lactam antimicrobial drugs like penicillin, carbapenem, ampicillin, and others. So if we summarize, there are different virulence factors in Staphylococcus. First of all, these are cell wall associated structures like Peptidoglycan, Capsule, protein A, clumping factor. There are also extracellular factors, toxins. This is hemolysin, leukocidin, Enterotoxin, exfoliating toxin. And there is also a coagulase activity. This is staphylococcus, dienase, phosphatase lipase, phospholipase, hyaluronidase, serokinase, protease, and others. So this is just one of the illustrations where we can see again here enzymes that are used for invasion. These are clumping factors, coagulase, staphylococcus, hyaluronidase, and lipase. All of these enzymes are used for invasion. Here there are binding proteins to the surface of the eukaryotic cell, immunoglobulin bound by the Fc portion. This is protein A here. And different exotoxins. About exotoxins we will start talking now. So, you know that there are exotoxins and endotoxins. Exotoxins are always released into environment, while endotoxins are released when the bacterial cell dies. Staphylococcus aureus produces many virulence factors, including at least 5 cytolytic or membrane damaging toxins, and these are alpha toxin, beta, delta, and gamma toxin, and also Panton-Valentine toxin. So when we say cytotoxic, this means that they are acting against the cells. They are either damaging the cells or destroying the cells. Alpha toxin is a hemolysin pore-forming toxin which lyses membrane, and it is inactive against neutrophils. So all of these toxins that we just listed, alpha, beta, delta, gamma, and Panton-Valentine, are cytotoxic toxins, cytotoxic factors. So what we see here on this picture, we see the visualization of the action of alpha toxin, beta toxin, and gamma toxin. Alpha toxin is on the blood agar, is forming grayish, greenish colonies, which means that the cells are partially destroyed, partially. Beta toxin destroys the blood cells completely. This is why around the colony we see yellow transparent halo, and gamma toxin does not give us any halo on the blood agar. Besides these cytotoxic factors, there are 2 exfoliative toxins produced by Staphylococcus, 8 enterotoxins that are designated from A to E and from G to I, and toxic shock syndrome toxin 1. Toxic shock syndrome toxin, abbreviation is TSST. So, exfoliating splits interdermal junctions. There are exfoliative toxins A and B, and their action results in staphylococcal scalded skin syndrome, SSSS. This is the abbreviation of this syndrome. Usually the infants and neonatals are affected by this syndrome. Staphylococcus scalded skin syndrome is a spectrum of diseases characterized by exfoliative dermatitis, is mediated by the exfoliative toxin. You can see here the face of this baby. And you see very intensive exfoliation on his face. The prevalence of toxin production of Staphylococcus aureus strains varies geographically, but generally it is less than 5% and 10%. So it varies between 5 and 10, but in general it is less than this. When we say the Staphylococcus aureus strains produce exfoliative toxins, and it is linked to geography. For example, in Georgia, you may have less percentage of Staphylococcus that are producing exfoliative toxin than in, for example, Armenia or Azerbaijan. Besides these toxins, there are also food poisoning enterotoxins produced by Staphylococci, and 3 subtypes of enterotoxin C have been identified as well. The enterotoxins are able to survive heating at 100 degrees for 30 minutes. So if we have some food in our refrigerators for a long time, and we think that after heating it will this food will become safe for us, it's a big mistake. No, if it is polluted with, if it is infected with Staphylococcus. And these toxins are also resistant to hydrolysis of gastric and genual enzymes. This is why it is very important to perform good cooking and sanitary actions when we are cooking. So the heat-resistant enterotoxin mediates Staphylococcus food poisoning, and the symptoms can be already seen in 2 to 6 hours. Usually these symptoms are self-limiting, but they are still unpleasant because this is nausea, abdominal cramping, vomiting, diarrhea. The foods that particularly are effective carriers of bacteria and its toxins, these are custard or cream-filled bakery foods, processed meat, chicken, salads with mayonnaise, ice cream, milk, dairy products. So you have to be cautious about this. So reheating is not effective. And these toxins are produced by 30 to 50% of Staphylococcus aureus strains. Exfoliative toxin is produced from by 5–10% of the strains, and these toxins are produced by more strains, from 30 to 50% of all Staphylococcus. Enterotoxin A is most commonly associated with this disease, and enterotoxin C and D are found in contaminated milk products. Enterotoxin B causes staphylococcal pseudomembranous colitis, so it is really not very pleasant thing to have the poisoning with staphylococcal enterotoxins. Now about toxic shock syndrome toxin and other toxins. This toxin belongs to the class of polypeptides known as super antigens, SA antigens. It is characterized by non-specific binding to the receptors and triggers excessive immune response, so that the person may die because of this immune response. Staphylococcus aureus strains produce several other extracellular biological active substances, including proteases, lipases, lysozyme, phosphatases, and others. Toxic shock syndrome is a superantigen which is capable of activating large number of T cells. It was found in the 80s of the previous century, and it was associated with the use of tampons by the ladies, but it is also known to be associated with the postoperative wound or soft tissue infections. The disease is characterized by high fever, vomiting, diarrhea, sore throat, muscle pain, which are symptoms of different diseases, including flu. Within 48 hours, it may progress to severe shock with evidence of renal and hepatic damage. A skin rash may develop, followed by desquamation at a deeper level, then in scalded skin syndrome. And the blood cultures usually in this case are negative, because toxin is spread in the blood. We don't see the bacteria, and we cannot distinguish them in the blood samples. That's why the blood cultures are usually negative, because all the symptoms are associated with the toxin production. Toxic shock syndrome toxin was called previously pyrogenic and exotoxin C and enterotoxin F. It is also resistant to heat and proteolysis, and the genes for this exotoxin are on the chromosome, are located on the chromosome. The ability of the toxic shock syndrome toxin is to penetrate the muscular barriers, even though the infection remains localized in the vagina or at the site of the wound where the operation was made. And this toxin is responsible for the systemic effects of the toxic shock syndrome. It is very dangerous toxin, because the patients may die, and the statistics showed in the previous century, when we didn't know that the tampons are associated with this disease, the mortality was between 5 to 10%. And this was because of the hypovolemic shock leading to multi-organ failure. We already mentioned that it can cause hepatic failure and other failures as well. Hypovolemic shock is an emergency condition in which severe blood or other fluid loss makes the heart unable to pump enough blood to the body. This type of shock can cause many organs to stop working. This is why we have to be very cautious about this. Now about the epidemiology of Staphylococcus aureus. Staphylococcus are everywhere, ubiquitous. All persons are carrying coagulase negative staphylococci on their skin, and there is also transient colonization of the moist skin folds with Staphylococcus. It is very common. Colonization of the umbilical stump, skin, and perineal area of neonatants with Staphylococcus aureus is also common. Staphylococcus aureus and coagulase negative staphylococci are found in the oropharynx of the people, in the gastrointestinal and urogenital tracts. Because staphylococci are found on the skin and in the nasopharynx, shedding of the bacteria is common and is responsible for many hospitally acquired infections. This is why it is absolutely necessary to screen the staff of hospitals for the carriage of Staphylococcus, and for this the nurse, samples from the nurse should be obtained. Usually the anterior nares of 20–30% of healthy people are colonized. Strains with increased virulence cannot be distinguished, so the community infections are endogenous usually, because this means that somebody within this community was a carrier, and hospital spread is on the hands of the medical personnel, and it is necessary to wash hands as often as possible. Outbreaks involve nasal carriers or workers with the lesions, and as we already mentioned, Staphylococcus aureus survives drying and survives on deformities, and that's why the disinfection should be done very frequently in the hospitals, and the disinfectant should be changed so that in this case we avoid the resistance to this or that chemical agent. Clinical manifestations. The clinical manifestations of some staphylococcal diseases are almost exclusively the result of toxin activity. We have seen already the food poisoning, the toxic shock syndrome. Sometimes the diseases result from the proliferation of the staphylococci, leading to abscess formation and tissue destruction. In case of cutaneous infections, for example, in case of endocarditis, pneumonia, MPM, osteomyelitis, septic arthritis. But staphylococcal diseases may be systemic as well, like, for example, toxic shock syndrome, when the toxin is absorbed into the blood and causes shock. Bacteremia also can be present. So this means that bacteria is present in the blood. Enterocarditis occurs when the bacteria attack the lining of the heart. Pneumonia is an inflammation of the lungs in which the alveoli and bronchioles becomes filled with fluid, and this is the result of the staphylococcal infection. Osteomyelitis is an inflammation of the bone marrow and the surrounding bone, also caused by staphylococci. Staphylococcal pyogenic infections are folliculitis, furoncoles, carboncles, bullous impetigo, panaricia, wound infections, mastitis, osteomyelitis, staphylococcal pneumonia. You can see here the skin infections caused by staphylococci. For example, this is bullous impetigo. These are bubbles, you can see, like swellings that can break and peel away. Most commonly we see them in newborns. This is folliculitis. This is a superficial inflammation of the hair follicles, usually resolved with no complications, but it can progress. You can see the pus is gathered here. So this is furunculosis. This is a boil, inflammation of the hair follicle or sebaceous gland progresses into abscess or pustule, and these are carbuncles, larger and deeper lesions created by aggregation of interconnection of different furuncles. This is ST here on the eyelash, and this is Panaricia. You can see it here, the redness. So this is because Staphylococcus it's a deep follicle. This is a furuncle, and you can see it goes deeper. This is a superficial follicle, this is a deeper follicle, this is a furuncle, and this is a carbuncle, and you can see the area of inflammation is much larger and deeper. For treatment, of course, we use beta-lactam antibiotics. This is oxacillin, methicillin, cephalosporin, different generations. Anti-staphylococcal antibiotics of the second choice are clindamycin, vancomycin, daptomycin, and others. For diagnosis, we use microscopy of the smears of the clinical materials that are stained by gram stain. Then we have to cultivate these samples on the solid media. This is usually blood agar, and also can be salt agar, because Staphylococcus is resistant to salt. Different biochemical tests, which is coagulase activity, catalase activity, should be performed, and phage typing is also giving us a response about susceptibility of Staphylococcus aureus strains to various temperate phages. Colonies on the solid medium are usually round, regular, smooth, slightly convex, quite large, 2-3 millimeters in diameter, after 24 hours of incubation. And here you can see, of course, this is the blood agar, and on the blood agar you can see that beta toxin was produced, and that is why we can see a halo around the colony. Most strains shows alpha hemolysis, which is partial hemolysis surrounding the colonies on the blood agar, and Staphylococcus aureus can produce cream yellow or orange pigment. This causes, here we don't see this pigment, but the name aureus, which means golden, Staphylococcus golden, comes exactly from the ability of Staphylococcus to form the pigment of yellow-orange color. Okay, this is the laboratory identification scheme. So we perform first gram staining. We should have the gram-positive bacteria clustered. Then we have to perform catalase test, and if it is positive, it is more likely that this is Staphylococcus. If it is negative, it is more likely that we see Streptococci. After this, after positive catalase test, we should perform oxidase test, and it should be negative if we have Staphylococci. Then we have to perform coagulase test, and if it is positive, we are having Staphylococcus aureus culture. If it is negative, this may be Staphylococcus epidermidis or saprophyticus. And after this, we are performing, so you can see that the negative coagulase test gives us the option. Either we have epidermidis, hemolyticus, or saprophyticus Staphylococcus. So you have seen this already. This is a coagulase test. So the growth of Staphylococcus aureus, for growth, it is a facultative anaerobic bacteria, so it can grow on air, All species grow best on nutrient agar and blood agar, and there is also mannitol salt agar, which is a selective media for Staphylococcus. As I mentioned already, it survives in salt. It can grow also in the range of temperature from 15 to 45 C, and the concentration of salt can be as high as 15%. This is mannitol salt agar. Staphylococcus aureus, when it is grown on mannitol salt agar, it changes pH of this media, and that's why we see the yellow color. It is not because of alpha or beta toxin, it is because of the changes in the pH. Staphylococcus epidermidis grows, but it does not change the color of this media. And here is a Novobiocin test. Staphylococcus epidermidis is sensitive to Novobiocin, whilst Staphylococcus saprophyticus is not. And this is how we distinguish between these two non-coagulase active staphylococci. Okay, we can stop here for a while, I think, and continue, or we can continue because we have quite a lot of slides still left. Would you like to have a break? 5 minutes? Okay. Only 5 minutes.